Hello everyone, I'm myself Dr. Dipti Kariya and today we'll discuss about erythropoiesis. Before starting my class, I would like to inform you all that you will get all my notes in soft copy and hard copy. To get the soft copy, you have to download the application and get the course. And you will get all my notes in the soft copy. And to get hard copy, you have to order my book, that is Physiology Notes, available on Flipkart as well as Amazon. Link for the Flipkart as well as Amazon and my application, they all are given in the description box. Now let us start our today's topic, that is formation of blood cells. Here the terminology hemopoiesis and other is erythropoiesis. Now hemopoiesis, that is process of development of all the blood cells, RBCs, WBCs and platelets. Okay, if it is uh, if it is development of RBCs, that is erythropoiesis. Development of WBCs, that is known as leukopoiesis. And thrombopoiesis or megakaryocytopoiesis, that is development of platelets. Okay. Now, theories of hemopoiesis. There are two theories. One that is monophylactic and second is polyphylactic. Monophylactic, that is most acceptable theory. According to this theory, different types of blood cells, they arise from single cell, and that is known as, you can see here, this one, single cell, that is known as pluripotent stem cell. Okay, and 75% of from this cells, they form myeloid series, means they produce WBCs, and 25% of this they form RBCs, that is erythroid series. And second is polyphylactic theory, that says that separate type of stem cells are there for different blood cells. Okay, now regulation of proliferation as well as maturation of blood cells. For that, it is regulated by various interleukins, and other is colony stimulating factor. Now, what is interleukin? This interleukins, you can see here, this is interleukin. They are hormone like chemical messengers, and they have amino acid sequence, and they are produced by our lymphocytes, macrophages and other body cells, endothelial cells, neurons, glial cells, activated T cells, fibroblasts, they all produce interleukins. Okay, second is colony stimulating factor. Here we have, I don't have the diagram. Colony stimulating factor, these are growth factors or hormones, and they cause appropriate single stem cells to proliferate, and in particular culture medium like in the soft agar, that forms colonies of the daughter cell, that is colony stimulating factor. Okay, now certain interleukins they play an important role in erythropoiesis, and they are interleukin 1, 6 and 3, and they act in the sequence. First interleukin 1 comes, then 6 and then 3, and they convert multipotent, or you can say uncommitted stem cells into committed stem cells. And in healthy individual, all types of cells in the peripheral blood, they are kept constant within narrow limit by feedback mechanism. It shows that rate of formation, release and destruction of blood cells, that is balanced. Now, sites of hemopoiesis. First, that is mesoblastic stage. Mesoblastic stage, that is in first 2 months, you can see here, of gestation. Here, mesoderm of the yolk sac, or you can say area vasculosa. Area vasculosa, that is the site of hemopoiesis. Okay, and erythropoiesis takes place inside the blood vessel, and this erythropoiesis stage, that is known as intravascular erythropoiesis. And remaining two stages, they are extravascular erythropoiesis. They are, one is hepatic stage, from the 3rd month of gestation till birth. Liver and spleen, they are main sites for the hemopoiesis. Okay, and after 20th week, it has been started earlier, but after 20th week of gestation, myeloid stage, means hemopoiesis begins in the bone marrow. So bone marrow becomes the site of erythropoiesis and hemopoiesis. You can say red bone marrow, that is active hemopoietic bone marrow. You can see here, this is the red bone marrow, and this is because it has marked cellularity. Okay, and yellow bone marrow, that is fatty replacement of the red bone marrow, and that is not active, and that will not play an important role in the hemopoiesis. Okay, so in adult, hemopoietic or red bone marrow, that is found in membranous bones like bones of the skull, vertebrae, sternum, ribs, sacrum, pelvic bones. Okay, and ends of the long bones here. So this is in adults. Okay, if suppose we are talking about children up to the age of 5 years, red bone marrow is present in all the bones. Okay, and 6 to 20 years, red bone marrows, that is present in long bones and flat bones or membranous bones. Whereas after 20 years, as we have discussed, ends of the long bones in adults and membranous bone, that is the site of erythropoiesis. Okay, now what is the differentiating feature between red and yellow bone marrow? We can easily say, we have already discussed, red bone marrow is red in color and it is active hemopoietic tissue, whereas yellow bone marrow is yellowish and inactive. The cellularity of red bone marrow is marked and it contains all the stages of developing cells, where the cellularity is very less and it is replaced by the fatty tissue. And this is present, as I told you, in the membranous bones and ends of the long bones, whereas in adults, except ends of the long bones and membranous bones, other all bone marrow, that is yellow bone marrow. Now, process of erythropoiesis, as we have discussed, all the blood cells, they are derived from the stem cells, and these stem cells are known as pluripotent hemopoietic stem cells. Pluripotent means they produce different types of cells. Hemo means blood cells or stem cells and stem cell, and they are also known as uncommitted stem cells, not committed to produce particular cell, whether they may produce RBC, WBC or platelet, anything. Then they are converted into committed stem cells, means now they produce one type of blood cell, okay, either RBC or WBC or platelet. So you can say that pluripotent stem cells, they are, you can say that pluripotent means they are not differentiated, okay. They can give rise to any type of cell. From that, they are divided in lymphoid stem cells. Lymphoid stem cells, they produce lymphocytes, and myeloid stem cells, they produce your either RBCs or WBCs, WBCs except lymphocyte and platelets. Okay, we will discuss in detail. Now, what are the features of progenitor cells? Progenitor cells, they have ability to give rise to grow, okay, and they can form colonies. So they produce colony forming units. Okay, three types of progenitor cells we have. Colony forming unit, you can see here, granulocyte, monocytes, from which granulocytes and monocytes they are produced. Colony forming unit, megakaryocytes, from which your platelets are produced, and colony forming unit, blastocyte, from which RBCs are produced. Okay, now how this hemopoiesis is controlled? There are various stages of erythropoiesis. RBCs, as we have discussed, they are developed from colony forming unit blastocyte, and that is converted into colony forming unit erythrocyte. Okay, now as we have discussed, this is just the same thing. Uncommitted stem cells, which are also known as pluripotent hemopoietic stem cells, which are converted into committed cells. Lymphoid stem cells and colony forming unit of spleen, from which this colony forming unit blastocyte and colony forming unit erythrocytes are produced. Okay, now you can see colony forming unit spleen, colony forming unit blastocyte, colony forming unit granulocyte, monocyte, colony forming unit megakaryocytes, we have already discussed. Now changes during, now we will discuss about this series, this one, colony forming unit blastocyte, erythrocytes, and from which RBCs are produced. So here, how this will go on? First of all, which changes they occur during erythropoiesis? What happens? Number one, size of the cell decreases as the cell grows. You can see this is larger one, this is mature is small. Second, nucleus gets condensed and disappears. Third, hemoglobin synthesis starts. You can see here hemoglobin is not present, now hemoglobin is produced. And fourth, because of presence of hemoglobin, staining property of the cell changes. Initially it was blue color, then it becomes red. Now, stages of erythropoiesis we will discuss. Let us first enumerate them. One, that is hemocytoblast, proerythroblast, early normoblast, intermediate, late normoblast, reticulocyte, and erythrocyte. Now, hemocytoblast, you can see here, this is the most immature cell here. The size is largest one, 19 to 23 micron diameter. Nucleus is large nucleus and occupies center. It is deeply basophilic cytoplasm, basophilic. Hemoglobin is absent and mitosis is +2 present. Okay? Then comes next stage, that is proerythroblast. Again the size slightly decreases but larger than RBC, 15 to 20 micrometer diameter, large nucleus with 2 or more nucleoli. Hemoglobin is still absent. Cytoplasm is basophilic because hemoglobin is absent. So blue color, mitosis is present. Then early normoblast, again size decreases here, 15 to 20, now it is 12 to 16 micrometer diameter. Nucleoli starts disappearing here. Chromatin gets condensed. Okay, cytoplasm is still basophilic because hemoglobin is absent. Okay, then comes your next stage. Let us repeat as we have discussed. First stage, that is hemocytoblast, proerythroblast, early normoblast, then intermediate normoblast. The size again decreases, the size is 10 to 14. Double then normal RBC, micrometer diameter. Nucleus is again basophilic and coarse. Okay, chromatin condensation continues. Hemoglobin starts appearing here. So the cytoplasm stains with both acidic and basic dyes, and therefore this cell is also known as polychromatophilic erythroblast. Okay, and mitosis is present +1. Late normoblast, again size decreases, 8 to 10 micrometer diameter. Nucleus is small and pycnotic, and it has just a moment. Dark chromatin material, cartwheel-like appearance. Hemoglobin quantity increases. Cytoplasm becomes, because of hemoglobin, cytoplasm becomes acidic and mitosis is absent here. Now, next is reticulocyte. It is just immature RBC, just before the RBC stage. This is reticulocyte. Here it has got reticular network. And what is this reticular network? That is made up of remnant of disintegrated cell organelle like Golgi apparatus, mitochondria, and other cell organelles. And this reticular network that is stained by supravital stains, that is brilliant chrysal blue. Okay, that stains the reticulum. Hemoglobin is now increases. Cytoplasm is acidophilic. Mitosis is absent. And in normal adult, these reticulocytes are 1%, whereas in newborn babies that is 2 to 6% normally present. Okay, now next is mature erythrocyte. This reticulum also now disappears, and mature erythrocyte gets the biconcave shape, and the size is about 7 to 8 micrometer diameter. And now this is your mature RBC. This is the morphology. Duration of erythropoiesis from proerythroblast to reticulocyte stage, it requires 5 days. Whereas from reticulocyte to RBC, the stage requires 2 days. Okay. Another important note here is bone marrow cells, hemocytoblast. They are few in number and they can be stimulated to form any cell in the body, and they are capable of completely replacing the bone marrow which is injected in the host, and that is your bone marrow transplantation. And how can we transplant? First of all, we have just to inject that bone marrow from the donor to the recipient. Before that, recipient's whole bone marrow that has been completely destroyed. Okay, so this is the applied aspect related to the topic. So this is all about stages of erythropoiesis. In our next class, we will discuss about factors regulating erythropoiesis. Again, if you wish to get all my lecture notes in soft copy, you have to download the application. You have to get the course from the application which is available in the Play Store. The application is Vesalius, and to get hard copy, you can order my book, that is Physiology Notes, available on Flipkart and Amazon. Okay, thank you so much. If you like my video, you can like it, you can share with all your friends, and subscribe my channel. Thank you.